Hey, this is an anime project submission from the pod 96 Romantic Google. We are the ManyManifolds. My name is Prakriti Nayak. I have just finished my master's in neuroscience from King's College London. I have no experience in computational neuroscience, so this is absolutely new for me. Hope you enjoy our project.
Hello, I am Narottam Singh. I took voluntary retirement from the post of scientist to investigate the nature of mind and the brain. I enjoy building automated home products and am an expert in both front-end and back-end web development. At present, enrolled in my third master in cognitive neuroscience at JSS Academy of Higher Education and Research, Mysore, India.
Hi, I'm Rishika. I am currently a fourth year undergrad at the Indian Institute of Science Education and Research, Pune. I am an undergraduate researcher at the Theoretical Neuroscience Lab and work on building models of insect olfaction.
Hello everyone, my name is Vaibhav Sharma. I'm currently pursuing my BTech in Information Technology from Netaji Subhash University of Technology, New Delhi. I'm in my first year and my interests include reading books, playing sports, and coding.
Initially, when we started this project, our question was whether we would see the motor activity encoded in different neural population represented on similar manifolds in the different regions that are implicated in the motor activity. But eventually, as we explored our data set, that is the Steinmetz data set, we landed on this question. So what happens in the data set is the mouse is given a visual cue and it has to move the wheel left or right in response to it. But in that left and right movement of the wheel is our proxy to motor output. But we want it uncoupled with the visual stimulus. So what we do is we stick to the open loop duration. Now that starts with the stimulation onset and ends at tone cue.
We are interested in motor control, so we only looked at the relevant motor regions in order to avoid other sensory confounders. Further, we only considered regions with more than 50 neurons and at least 2 mice in order to ensure reproducibility and some degree of population coding. Only the open loop duration between the stimulus onset and go cue.
Here in the step 1, the population spike code part of the project, we fitted a GLM. The work involved sampling random neurons 50 and 100 trials in the relevant brain region, pre-processing them so that only the relevant data from the onset of the stimulus to the go cue is fed to the GLM. Since preceding neural spikes can influence the present, we first found the optimal value of D for different brain regions and then trained GLM with the optimal value of D. To remove any selection bias, the whole process was repeated multiple times on randomly selected neurons and trials.
In our second approach, which we are calling the latent information code approach, we wanted to extract latent dimensions from our spiking data. Our first option was to use principal component analysis, but that did not work out because we did not have a general way to smoothen the spikes. So we adopted a more advanced approach, the Gaussian process factor analysis method. And from the neural modes that we extracted, we tried to regress the motor output. Due to the nonlinearity of the system, it did not work out that well. So we converted it into a classification problem and tried to use linear discriminant analysis to discriminate between left and right motor output.
Each brain region has different optimal temporal depth. This gives an insight into the temporal response of different brain regions. And not all brain regions contribute equally in the motor control task.
In the latent information code approach, we found evidence that suggests latent neural modes of different areas don't separate motor output equally. Certain regions, such as the primary motor cortex, were able to separate left versus right decisions in a low dimensional population activity space, while other regions, such as MD, failed to do so. We quantified this using a balanced accuracy and a weighted F1 measure to deal with the imbalance of the classification labels in our dataset.
We had lots of fun experiences and it was awesome. We learned a lot and Jason, our mentor, was really helpful. Finally, do check out our code, results, and more details on our GitHub page. Thank you.